food, drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. By just telling everyone a little bit about who you are and what your lab studies?
Sure. Yeah. So, I run a pancreatic beta cell biology laboratory at the University of Michigan. I've been here for a little over 11 years. We're really interested in the regulation of mitochondrial quality control in beta cells and how that relates to the pathogenesis of both type 1 and type 2 diabetes. And then we try to use our basic laboratory to try to bridge questions that are relevant for understanding the pathogenesis of diabetes and to hopefully learn insights that can be valuable for translation to the clinic.
Mm-hmm. And these pancreatic beta cells are obviously key for understanding diabetes. So let's just give everyone a very, very quick, very basic crash course in the pancreas and the beta cells in particular. What is the pancreas basically doing as an organ, and how are beta cells working at a very basic level in a normal, healthy individual?
Yes, great question. So I like to think of the pancreas as actually two organs together. So you have 98% of the pancreas, that is what we call the exocrine pancreas. That's the parts of the pancreas that are responsible for making digestive enzymes that will be fed into the gut to help you digest food, like fats, especially in your food. Mm-hmm. And then there's a whole, interconnected series of ducts that collect those digestive enzymes and then get them into the intestine. And then 1% to 2% of the pancreas are called the endocrine pancreas. That's what we study in my lab. And the endocrine pancreas is primarily organized into these structures called islets of Langerhans. And they're essentially microorgans that are made up of many different cell types that are kind of sprinkled throughout the pancreas. And the different cell types in the islet release hormones that have a lot to do with the regulation of glucose levels in the body. Mm-hmm. You mentioned the beta cells. The beta cells in a human comprise maybe 40 to 50% of the cells in the islet. And they make insulin, which is the hormone that is really important for helping us metabolize glucose, getting glucose into our cells so it can be metabolized. And then there are 5 to 6 different endocrine cell types in the islet. But the other one that people think about a lot and hear about a lot are the alpha cells, and those cells make a hormone called glucagon. That's also about 40% of the cells in the islet, and glucagon does a lot of things, but most of the time people think of glucagon as a hormone that helps you respond to low blood sugar, starvation, and the like. So we have about this is again approximate. We have about 1 million islets in the pancreas of an adult human. And in a normal person, when you're eating a meal and you have nutrients come into your body, especially carbohydrate, but also amino acids and fat, those fuel stimuli raise your blood sugar, especially the carbohydrates as they're metabolized, and that elevation in the blood sugar stimulates those beta cells to release insulin to go into the bloodstream so you can metabolize that sugar.
Mhm. So we can think of the pancreas as having two main parts. The bulk of the mass is actually devoted to making digestive enzymes. So those enzymes are manufactured there, and there's some kind of distribution or piping system that then gets all those enzymes shipped out to our gut, where they actually do the work of chewing up our food. And then a smaller portion of the pancreas, but a very important portion, is this endocrine portion. And this is where we start talking about hormones and insulin, and we get into the diabetes biology and all that stuff.
Exactly. Yeah. And insulin is not released into a duct network, right? So there's these islets are really vascular. They have a really, really substantial blood supply. 1 to 2% of the cells, but they have like an outsized vascular flow relative to the rest of the pancreas. So the islet cells, the beta cells, alpha cells, they just release their hormones right into the circulation, and then they go
Mhm.
and do the job of metabolizing nutrients.
Mhm. And do they have outsized vasculature? Because the very nature of what's going on here with insulin is to sense and respond and help regulate blood glucose levels. So I'd imagine it's got to be a very quick and
Yeah,
sensitive mechanism there.
absolutely. Yeah. I mean, I think, these are cells that need a lot of nutrients, but then there are also cells that need to be rapid responders to what's going on in the body. So, I think those are two prominent reasons why they're really vascularized cells.
Hmm. And then we usually talk about two types of diabetes, type 1 and type 2. I know that you also have a personal connection here. So, do you want to talk about that a little bit, how you got into
Sure.
this and how that connects to type 1 diabetes and what that is? And maybe we'll talk about
Absolutely.
that for a bit before probably spending the bulk of our time on type 2.
Yeah, 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 absolutely. And thanks for giving me the opportunity to talk about that. Yeah, so I was diagnosed with type 1 diabetes when I was 5. So, not to give away my age, but like we're approaching 40 years that I've had diabetes. And I was born and raised outside of Cleveland, Ohio, a small town called Youngstown, Ohio. And that kind of, changed everything for me. So, I was just normal kid, like to eat, snack, maybe a little too much. And then all of a sudden I had 2 weeks of like this increasing kind of just not feeling well.
Hmm.
The story that I prominently remember, I don't remember much from that age, was we were at a wedding, a family wedding, and I kept going to the bartender to ask for Coke or Pepsi or whatever,
Hmm.
and because I was thirsty. So I would have a Coke or Pepsi, I would drink it, and then I'd run to the bathroom, and then I'd come back, and then my dad would see me going back for like more, and he
Yeah,
was like,
yeah.
"Stop drinking. Why are you drinking so much Coke?"
Hmm.
And I was like, "I'm thirsty. I don't know what to
Were
tell
you
you."
thirsty for like a water or were you thirsty for something sugary?
Probably a little bit of both. I will say that I can't give you like the greatest play-by-play of the inner
Yeah,
workings
yeah.
of the 5-year-old mind, but like for some reason in my head it was Coke or Pepsi that I really wanted. Could also just be that I was a kid at a wedding and was like, "Oh, I can have Pepsi
Yeah.
whenever I
But
want."
you were unusually thirsty.
Oh yeah. Yeah, absolutely. And then my folks were like, "Wait, there's something not right here."
Hmm.
And that evening, I was still using the bathroom a lot. And my parents were kind of putting their heads together what was wrong. And then, shortly thereafter, they took me to the hospital, checked me out. My blood sugars were through the roof. I got admitted to the hospital. Um, and and then, that kind of just that was a world-changing event for me. So some of this is recollections that are shared with my folks, but they just said, I was really, really interested in what was going on as much as a 5-year-old could be. I really was back then when you got diagnosed with diabetes now. So I should also say I'm a physician who takes care of people with diabetes too. When you diagnose folks with diabetes now, oftentimes they're in and out of the hospital very quickly, sometimes hours.
Mm.
But back then, there was this belief in the mid 80s that, you admitted the patient to the hospital and you had like intensive education and training and all these things. So I spent 2 weeks in a children's hospital in my hometown.
Oh, wow.
And yeah. And so, I was in the ICU for a couple of days because my sugars were really bad. But most of the time, I was just in a pediatric floor. And so I started expressing an interest in like helping out with the staff, figuring out what else was going on. I just
Mm.
kind of that lit that fire of interest in, diabetes and medicine and the like. So that kind of changed everything. And I became very interested in knowing more about this disease. And I don't know, it's probably the way I was raised or whatever, but like maybe being a Midwestern kid, like if you have a problem, you have to fix it yourself. But
Hmm.
I very much kind of drilled into this, and I don't think it came from externally. I think this was just me. Diabetes was my disease. I wanted to figure out why I had it and
Yeah.
see if I could fix it.
Hmm.
And that's kind of just been the fabric of my entire life.
Wow.
Um,
And is five so for type 1 diabetes, what people often call, we used to call childhood diabetes because you diagnose it at a relatively young age.
mhm.
Is 5 years of age a normal age? What's sort of that distribution look like?
Yeah, I mean, I think that typically it's somewhere between the 5 to 12 window. So I was probably on the youngish end, but like that's prototypically in the old days like juvenile onset
Yeah,
diabetes. Like I was on the earlier kind of window of that, but still like kind of in this typical place for type
I see.
1.
So it's truly a childhood onset. It's not an adolescent thing normally.
Yeah, I would say if you like looked at the numbers in detail and said, what is the biggest population? But the thing that we know now about type 1 is that you can get diagnosed with type 1 throughout your lifespan. So, I take care of people with type 1. We have folks come into the clinic that get diagnosed with type 1 in their 20s, 30s,
Hmm.
40s. Um,
Is that because they had it all along and it was a failure of the diagnosis, or to pick it up, or are people getting it at later ages as well?
yeah, so that's also a great question. I think that the more we learn about type 1, there are a few things. So I think historically there were people that would get diagnosed with diabetes as an adult, and then the assumption is, oh, it's got to be type 2 diabetes
I
because
see.
you're an adult. But when you rigorously study some of these folks, it doesn't change the fact that their blood sugars are high, but it
Right.
just changes the assumption you made when you met them. But when you measure production of your own insulin, when you measure autoantibodies to see if there's an immune attack, then it starts to change the picture.
Hmm.
And then make you think, oh, there could be type 1
Hmm.
here. Now we know you can diagnose, there's clearly type 2 that gets diagnosed in childhood as well. So I think the age barriers have kind of slowly drifted
Disintegrated.
away.
Yeah.
And while you get more type 1 in childhood and more type 2 in adulthood, the age barriers I think are kind of starting to change. And we just look at like what's the clinical picture? What
Hmm.
does the data tell us about what the patient might have? And
I see.
and then we have to kind of do our best to try to fit people into a box, which is also sometimes challenging.
Yeah, I would imagine. I mean, clearly, there's always been a kind of bimodal distribution here, right? There's a cluster of people who get it relatively young, often in childhood. Those are almost always type 1. The people who get it closer to adulthood are usually type 2. So that probably gave rise to the presumption most of the time that if you were an adult, it had to be type 2, even though not
Right.
necessarily, and vice versa. But then over time, I think there probably was some true blurring here because I suspect just as type 2 has become more common at younger and younger ages due to diet and lifestyle issues that happen earlier in life, I would imagine that maybe the autoimmune side of this or type 1 can happen later and later in life as history has progressed because there's probably things in our environment that can instigate that at later times than they used to.
Yeah, I think that's a very fair way to put it. And some of it is also changed based on the evolution of our understanding of the diseases.
Mm-hmm.
So as you said, in the mid 80s, there was no type 1 or type 2. It was juvenile onset and adult onset.
Hmm.
And then we learned, oh wait, you can get this picture in adulthood. Okay, we need to change the names. And then in the late 80s and early 90s, they started to call it insulin dependent because there were adults on insulin that
Yeah.
didn't look like a prototypical type 1. Um, and there's non-insulin dependent diabetes. Well, and then some folks thought, well, that doesn't work either because like everyone on insulin doesn't have the same type of diabetes.
Yeah.
And so then later it evolved into type 1 diabetes, which again, most people think maybe some degree of autoimmunity, and type 2 diabetes, maybe some metabolic problems. But I can tell you too, like personally, I have problems with those names too, because
Right.
we're learning more about these diseases. And so, maybe it's a lot of, discussion about nomenclature, but I think as we learn more about these diseases, it changes the way we view them and what causes them and
Yeah.
and what goes into it.
Can you give us a brief overview of what we know on the pathogenesis of type 1 diabetes today? My understanding, someone who pays attention to this stuff but is not in the field and not, I'm not up to date on the literature in this particular domain. I think historically the my understanding has been that type 1 is an autoimmune thing. Your immune cells attack your own pancreatic beta cells and destroy them, and then they're destroyed, and then you can't make your own insulin. Is that the full picture, or is that incomplete?
So that is the perfect general distillation. And that's what I was taught in med school. And that's what our medical students learn here. And that is the view. But I give a lecture to our med students and then I kind of shake things up because it's not the full view.
Mhm.
Um, so what we're learning from the genetics, the pathology, the histologic study of type 1, the natural history, is that yes, there's an immune component, but that's not it. There's also an intrinsic beta cell problem that occurs in type 1.
I see.
There's also some evolving work that the exocrine pancreas at the beginning, which I was like, oh, that doesn't do anything. Forget that. There also seems to be some evidence that there's problems in the exocrine pancreas in type 1 that might agitate the
I see.
islet immune interface and also contribute to the disease. So I think of type 1 really, if you want to be fair, as a multi-organ disease.
Mhm.
It's a disease of sick beta cells and then sick immune cells and potentially a sick pancreas writ large that then have these conversations that lead to overall beta cell loss. The
I
the
see.
other interesting thing is the beta cells themselves seem to not always fully disappear in type 1.
Hmm.
If you look under, a microscope, and we've had, the field has had the fortune to study autopsy specimens, or pancreas sections of folks that had type 1 and passed away and donated their organs for research. And a lot of these folks, sometimes people up to 50 years with type 1, still have detectable beta cells. And the immune attack of these, especially new onset type 1s, the immune attack, relatively speaking, is kind of underwhelming.
Mhm.
And you have like this patchy pattern where you'll have like some islets and some beta cells surrounded with like a small number of immune cells, and then other islets, there's no immune cells at all.
Hmm.
So, it started to kind of change our view that it's not just all about the immune system. There's probably more to it.
Mhm.
And I'm happy to
Yeah.
go into detail on what are all those things too. But I think it's fair to say at this point that, if you're talking to someone that isn't in the field, that doesn't know much about diabetes, and it's like, give me like two
Mhm.
words, autoimmune disease. Yeah, it's not wrong. But I think if you say,
Yeah.
well, it's autoimmune disease, it's autoimmunity plus these other things,
Right.
that's
Right.
more accurate.
So there's probably some intrinsic component here. Basically, some people are just born with beta cells that aren't quite functioning in the fully normal way. Maybe the other part of the pancreas is sort of leaking enzymes over there, and that's aggravating something too. And then somehow, some way, the immune system gets triggered inappropriately to attack these cells. What do we know about Do we know what those immune cells are recognizing? Are the beta cells in type 1, people with type 1 diabetes, are they producing some kind of molecule that the immune system is mistaking as a pathogen or something like this?
Yeah, I will give the caveat that I'm not an immunologist, but I do know my immunology colleagues. I mean, this has been a focus for a long time, is really identifying those antigens that the immune cells respond to.
Mm-hmm.
Certainly, there are hybrid peptides and abnormal forms of insulin that seem to be immunogenic. But the discovery of the diversity of these probable abnormal peptides, likely coming from a beta cell source, um, that kind of miseducate the immune system or are educating just fine, but then you have abnormal cells in the thymus that normally would, we call them autoreactive T cells. They normally would be cleared and you wouldn't kill your beta cells. But for some reason, these autoreactive T cells are hanging around and then
Hmm.
they go and attack the beta cell. So,
Yeah.
I think it's pretty safe to say that like hybrid forms of insulin and insulin peptides are a part of it, but there's probably more to the story and the challenge is kind of how you identify all of those very unique peptide antigens, but really phenomenal immunologists trying to figure all that out.
Mm-hmm. Have the rates of type 1 diabetes changed over time? Has it gotten better or worse?
That is again, I'm so glad you asked these questions. Yeah, so there's this assumption, right, that diabetes is on the rise, and it is worldwide. And then the next thing that people say is, oh, well, we're all gaining more weight
Mm-hmm.
and it's all type 2.
Right, right.
But
That
if
would
you
be the
look
natural
at,
thought.
right, and that's not wrong that type 2 diabetes is
Yeah,
on the rise. And
yeah.
obesity rates worldwide are on the rise, and those things are not wrong.
Oh boy, what are
But
you about to say?
type 1 diabetes incidence is also on the rise. It's not maybe on
Wow.
the steepness of the curve,
Right, right,
not the same
right.
as
But,
type
you know,
2.
but that would say, if hopefully I'm not caricaturing too much. If sort of one of the old views was this was like a fully intrinsic, you're born with it type of thing, you would not expect to see this. So that implies that something in the external world is probably causing this rise.
Absolutely. And then if you look at these, historic, but I think very powerful monozygotic twin studies, they give you this idea that there is an environmental component to type 1 and a genetic one. So, the old studies suggest that if you have one twin that gets type 1, the other identical twin has a rate of getting type 1 at about 50%.
Mm-hmm.
There are some people that think that we might have underestimated those because maybe we didn't study the other twin long enough, and it could be even as high as 70%.
Mm-hmm.
But if it was fully genetic, then it would be 100%. The other
Right.
twin would get type 1. It's not.
Yeah.
So
It's still
there
way
is an
more,
environment
it's still
component.
largely genetic because, or there's a huge genetic component because it's way more than the background rate, but it's
Absolutely.
definitely not 100%.
Right, right. And is the environmental component part of the reason why the incidence is rising?
Right.
It's very possible.
Hmm.
And that's also, there are lovely studies like, the Teddy study and Daisy study, these studies that are trying to figure out like, okay, well, we kind of know, we at least know the genetics. We don't know what the genetics are doing. We know the genetics have been profiled.
Right.
What do the genetics mean is a whole other kettle of fish. But the environmental triggers are still, really intensely being studied,
Mm-hmm.
and
Mm-hmm.
there's a lot that we really don't know.
Yeah. And I mean, we don't necessarily need to get into it. It sounds like it's still fairly mysterious, but immediately that makes me think of just the general rise in autoimmunity that we've seen for some number of decades now. This would seemingly be related to that. It's just a piece of that puzzle. Why are our immune systems developing and being trained differently over time?
Yeah, if I had the answer for that question, then I probably would be living on some fancy private island or something
Yeah,
like
yeah,
that, right? But like, we don't know. There is some, I think good evidence that certain, viral proteins are found in islets of folks that have type 1.
hmm,
There are certain, sophisticated immunologic studies that show viral proteins in the islet or in these immune cells that are attacking islets. that make you think that, certain viral types might give rise to it as an environmental trigger. Um, I think that most people would say that's not unreasonable to think that, but like if you ask somebody, well, tell me the specific virus type
Yeah.
or which viruses, then it gets a little murky. So
Yeah.
some folks think there's good information on enteroviruses. Coxsackie virus has been like linked to type 1 going back 30, 40 years. No one has disproven Coxsackie virus involvement. I think people still believe that that's a possibility. And then, there are other environmental things that people have discussed, but like, how strong the evidence is, I don't really know. Like cow's milk antigen was one of them for a while. And it's like, oh, when did you feed your young one milk? When didn't
Right.
I?
Right.
I don't know how strong that data those data are, but I know the folks that are doing Teddy and the like, they're trying to find strong links in the environment too, because they realize it's more than the genetics, even if the genetics are a big part of it.
Yeah. Yeah. So the bottom line is type 1 diabetes is on the rise to some extent. It's not as common as type 2. It might not be rising as fast as type 2, but it's not a purely genetic phenomenon.
Correct.
Okay. So, and just to put an end to this section, so the sort of end problem with type 1 is that the beta cells get destroyed to some extent, and so the body's capacity to actually make and secrete insulin is what is compromised.
Yeah, absolutely. And, so our patients with type 1, require lifelong insulin replacement therapy. And, we live in an era now where you can get insulin, from pumps but you can take in, the old school way is you take a shot, under the skin and different forms of insulin and checking your blood sugar. And that's still the backbone of therapies ever since insulin was discovered over 100 years ago.
Mm-hmm.
But we're moving towards an era, hopefully in the next few years, of maybe more automated insulin pump technologies, and then even into the science fiction, hopefully science fact of maybe stem cell replacement of insulin producing beta
Mm-hmm.
cells.
And
Um,
do those beta cells for in type 1, are the beta cells constantly trying to regenerate themselves? Are they dividing and then the immune system just keeps attacking them, or do they just sort of permanently go away and they don't regenerate?
Another really good question that it's hard to answer because when we get the tissue from a deceased donor, it's hard to know, right?
Mm,
Was this like a new beta cell or was this an old one that managed to like hide and evade all these years?
yeah,
There's been a lot of speculation about like what is the origin story of those beta cells that are still there.
yeah,
There are some folks that believe both, that they were, they might be lost their identity so that the immune system couldn't pick them up, like a pseudo camouflage of sorts. There's some good evidence to support that. There's also some evidence that maybe there is some regenerative signals. But again, is it one or is it both? Is it a little bit of everything? It's hard to know, but it does give some ideas in terms of like maybe things that you could do. Is there potential for endogenous regeneration? Would that be sufficient? Who knows? If you were to try to stimulate endogenous regeneration, do you need to camouflage those cells because the immune system is still around?
right,
These are things that we're actively trying to figure out, and I don't think people quite know, mostly because you can't, and you rightly shouldn't be able to, you can't do that like from someone with diabetes, right?
yeah.
This is an organ that you can't really study in real time.
Mhm.
So
So turning to type 2 diabetes, I'll kind of let you steer us in the beginning here. What is type 2 diabetes? Are there multiple subtypes? How do you start to think about this today in terms of what type 2 is and what the sort of core dysfunction leading to it is?
Mhm. So, I always give everyone the preface that yes, I'm a beta cell biologist. And so, a lot of people are always like, well, the thing that you study is center to your universe. That must be why you study it. Um, but as a kid with diabetes, I got to beta cells because I thought they were core to the disease and not necessarily because like I happen to work in someone's lab and I'm carrying the flag, right?
Mm.
So I'm trying to make sure that people don't think I'm biased, but everyone will make their own decisions. I will say this. Whenever people ask me about the origin of type 2 diabetes, like obviously metabolic overload, obesity, is definitely part of the equation. Like, and no one will dispute that. But there are some interesting peculiarities when you look in the clinical space that make you wonder, like, why do you get type 2?
Hmm.
So here's a couple of them. One of them is if you look in the US, approximately 1 in 2 people are considered overweight. Okay. If you look in the US, 1 in 10 people have type 2 diabetes or have diabetes, and the vast majority of them have type 2 diabetes. So here's the interesting thing. The majority of people that are overweight or that have type 2 diabetes are also overweight. So I'll do a quick distillation of the math and say, all right, if 50% of the population is overweight, 10% have type 2, then in a reductionist way, 1 out of every 5 overweight people have type 2 diabetes,
Hmm.
right?
Yep.
But turn that number on its head. That also means 4 out of every
Right.
5 people who are
Only, you could
overweight.
say only 1 in 5. Why isn't it 5 out of
Right.
5?
Exactly.
Yeah.
So 4 out of 5 people that are overweight don't have diabetes. And then you could say, oh, well, you haven't studied them long enough. But like these are like long longitudinal like looks
Yeah.
at our population. It's not these things are pretty constant.
Hmm.
So then what is up with those 4 people that don't get diabetes versus the one person that does? And this
Yeah.
is where I say the beta cells matter. If you're overweight, you have obesity in all the cases. The folks with type 2 diabetes have a beta cell problem.
Yeah.
They have beta cell failure, burnout, loss of function, however you want to term it. And so, and then really elegant work that was done in Seattle, in the late 90s, early 2000s, kind of really solidified this this effect in studies in humans, that the folks that get type 2 diabetes develop an intrinsic beta cell problem, that their beta cells can't compensate appropriately for their relative degree of insulin resistance that's driven by their relative degree of obesity.
Hmm.
So this is a beta cell disease.
Yeah. I guess
But
another way of saying all of that is obesity in itself cannot possibly be the driver of type 2 diabetes.
right, exactly. Now I'm talking garden variety, big picture, right? And you
Yep.
can definitely
Yep.
cite examples of extreme obesity syndromes where people will get diabetes. But if you look at garden variety type 2 diabetes, there is a beta cell problem that is tipping the person over the edge from pre-diabetes to diabetes.
Hmm.
And there's also a couple other very interesting things. Again, I distilling large quantities of work done. Um, the vast majority of the genetic loci associated with type 2 diabetes, and there are a lot, there's probably 400, maybe up to 500 different loci, many of which have very small contributors to the disease.
Hmm.
But the vast majority of them encode products that have key beta cell functions.
I see.
So the genetics kind of push us to the beta cell. And to me, I think one of the most fascinating things, and this is, I regularly cite this study when I talk to students or if I go on the road to give a talk to a wider diabetes audience. A study done in the UK led by a guy named Roy Taylor. Um, this is done in the pre-Ozempic, era, but it was published in 2018, and they took early onset type 2 diabetes patients who were overweight and either with intensive diet or surgery, treated these patients to bring their body weight into the normal body weight range. They were no longer overweight. And here's the fascinating thing. The patients split into 2 groups about 50-50. 50% of the people, their blood sugars fell into the non-diabetic range.
Hmm.
So you could call them responders to the weight loss.
Yep.
The other 50%, they still had diabetes.
Right.
They were
Okay.
non-responders.
Hmm.
Both groups had identical, liver fat reductions, pancreas fat reductions, weight loss was similar amongst the groups. All of these things were the same. The difference between them, the responders and the non-responders that stayed with diabetes, the non-responders didn't recover beta cell function, didn't
I see.
recover the ability to secrete insulin to a glucose channel.
I see.
So,
So it comes back to the beta cells again. And, just to summarize what we've said so far, it sounds like what you're saying is so obesity is neither necessary nor sufficient for someone to have diabetes. You can be obese and not have diabetes. You can also be lean or non-obese and have diabetes.
right.
So there might be common drivers, which is why there's a correlation. There's a relationship between obesity and diabetes, but it's certainly not one-to-one.
Right, precisely. And that's not to say that if you have type 2 and you're overweight, that losing weight and eating right isn't going to help you.
Right.
That would I'm not that would be an insane thing to say. And in our clinic, we definitely advise like lower weight, exercise, diet, reduce carb intake, all those things. That is literally the backbone of type 2 diabetes treatment. But the thing that we always remind our patients is that's a start. And if you're lucky, yes, maybe it'll get you off all your diabetes medications, but there's a good chance it won't. And then when it won't, then we talk about other medical approaches to treat you.
So let's talk about beta cells and insulin a little bit. So type 2 diabetes, to what extent, this is something that I'm a little confused on. I don't know if this is known and I'm just ignorant or this is still being worked out. So to what extent is type 2 diabetes an issue of insulin secretion by the beta cells versus insulin resistance elsewhere?
Mm-hmm. So, we talked a little bit about diabetes subtypes too. And I will say there is this evolving look at type 2 diabetes to break it down into subtypes also.
Yeah.
I would have to remind myself there are these like 4-letter acronyms for the different subtypes of type 2. I'm still trying to get my head around them. They're very helpful when we talk in the molecular space. I will say I haven't gotten to the point in my clinic where I'm breaking the patients down into these categories. But there are folks with type 2 that definitely don't have autoimmunity. And, they're definitely like severely insulin deficient. And then there are folks with type 2 that have severe insulin resistance as well. And they're part of that whole subgroup. And then those two are probably like smaller components of type 2. They're not like tiny. They're there. But then probably the bigger ones are kind of the milder obesity related diabetes. And then probably the most prominent one is like the milder like age related diabetes. Those are the two more prominent forms. So in the garden variety, in those two subtypes, you get some insulin resistance. It probably, and there's certainly some correlation with continued weight gain, but to most degree, the insulin resistance tends to plateau. And then you get some degree of beta cell dysfunction, but in large part, most of the time, diabetes is a progressive disease. So, you see someone with garden variety type 2, you start them on, say, a medicine like metformin, and then you follow the person for 10, 20 years. And then in 20 years, they're on like multiple drugs and maybe an injection or maybe even on insulin. And that progression is likely related to like continued beta cell loss with time. So they're both factors. The insulin resistance probably starts first, early on in the disease process, and the beta cell is probably trying to figure out how to deal with it. And then at some level, at some point, it just can't anymore.
I see.
Can't make more insulin, can't figure it out anymore.
I see. So in some sense, in the abstract, maybe loosely, we could, there's insulin resistance to some extent somewhere. The beta cells are trying to keep up with that, so to speak, and they kind of get burned out in some way, shape, or form.
Yep.
So
now I will also say there's also to be fair, because I have a few friends in the field, and if they watch this, they'll like maybe send me a note and get disappointed in me. I will say, there is a community of folks as well, and it's not like one or two people. There's like a decent number of folks in our field
Hmm.
also. I would say this isn't the commonly held belief, but to be totally fair, there are some people that think that insulin resistance is actually its origin come from the beta cell too. So that
Mm.
some people think that actually like an intrinsic problem in the beta cell is like over release of insulin that then starts the cascade of peripheral insulin resistance everywhere and then starts this back and forth feedback that then leads to eventual beta cell loss and diabetes diagnosis.
Hmm.
So it may not just be as simple as like, oh, you had too much junk food and then you just got obese and then the adipocytes and the muscle cells and the liver were wearing your beta cells down. There are some people that think, oh, there was a messed up beta cell hypersecreting early on that set off this insulin resistance as well. So
Hmm.
that I just want to be totally fair to say there are some folks that think that the origin of insulin resistance actually itself might be with the beta cell too.
Hmm. And when we talk about insulin resistance, are there any patterns of so many tissues are insulin sensitive, they have insulin receptors. My understanding is insulin resistance is often tissue specific. You might have some organ or tissue systems that are insulin resistant, some that are insulin sensitive. These can change over time. Is there Are there any common patterns where certain organs tend to become the most insulin resistant early, or is this does this tend to start with the liver or start with the whatever? What do we know there?
Yeah, so now I'm a little bit out of my comfort zone because I'm a beta cell person. I will say that it is believed that the muscle is the one that takes up and processes the vast majority of glucose in our bloodstream. And
Yeah.
so insulin resistance from the muscle probably exerts like a very prominent effect on overall systemic insulin resistance.
Yeah. Yeah.
But I think that the usual suspects of like the cell types that we think of that are most prominently reliant on insulin for glucose uptake are muscle, white adipocytes, our normal fat cells, and the liver.
Mhm.
And and in the case of the liver, it's not insulin related glucose uptake, it's more insulin related, glucose processing. But in the adipocytes and in muscle, it's insulin related glucose uptake being,
Yeah.
prominent. I'd say muscle does most of the glucose
Yeah.
metabolism.
Just a natural. Yeah. I mean, you would naturally think that's going to play an outsized role because yeah,
Mhm.
I mean, it does play an outsized role. It's
In terms of like, how does it, do some come and go and some have differing kind of features during the process? I have to plead my ignorance with that.
yeah.
It's very possible that they do, and I just don't know.
Yeah. So there's a lot of complexity here. Obviously, I'll let you kind of steer us, but I'm just going to start down a track and we'll see if this gets us back to the beta cells or whatever. But one of the so there's a question in my mind of obviously there's a huge lifestyle component that probably has some interplay with some amount of intrinsic factors that different people have that can lead to type 2 diabetes. People often talk about diet, and obviously that's somehow ties into this. In my mind, I like to separate dietary food components. So things like we're supposed to consume food, the foods and the things that we're consuming, that we're supposed to be consuming. And then things we might be consuming that we're not supposed to be, things like endocrine disruptors and chemicals and stuff like that. When we think about type 2, well, let me just put it this way. One of the sort of common cartoon caricatures out there for where type 2 diabetes comes from is, well, you're eating a lot of sugary, starchy foods, you're eating too many carbohydrates like added sugars, your cells are overloaded with too much sugar, and the insulin resistance is coming in response to that. And type 2 diabetes is ultimately sort of coming from eating too much sugar. That would be one caricature that's out there. To take us, where would you take us from that?
Yeah. Well, I mean, look, it's easy, I mean, this is we all try to like understand the world in simple ways, right? That's the
Mhm.
way that we are kind of wired. Um, is that an easy way to apply for everybody? No, of course not. But I mean, certainly like caloric overload,
Hmm.
and if you think about, body weight being a balance of caloric intake and weight loss mattering, then certainly there's a component to that. But as you mentioned, there's also an intrinsic component. Like some people have much better resting metabolic rates. There are some people that are more prone to obesity than others, right?
Hmm.
There's a genetic component to obesity as well. It's not always as simple as like, oh, you ate 1,500 calories a day and you ate 1,500 calories a day, so neither of you should be overweight, right? Like,
Yeah.
there's more to it than that, right?
Yeah.
So, yeah, I mean, that caricature of like, oh, it's just like you were sitting, with like
You
whatever.
ate too much sugar and didn't move enough.
Right, you're eating donuts and Cheetos and
Yeah.
whatever, and that's why you have type 2 diabetes and you're overweight and like you were eating, grilled chicken salad every day and you didn't get it, right?
Hmm.
And like, again, that's a lot of oversimplification. And
Yeah.
so I think you have to give room to the idea of like there are intrinsic things that regulate our weight, that regulate our appetite. There are probably certain foods that are going to be more metabolically problematic in some folks than others, but
Hmm.
certainly carbohydrates are, getting attention, which they should. Because they're making those beta cells work. They're very prominent stimulators of those cells. They're very effectively turned into, lipid and stored in adipocytes and promote weight gain. So like certainly carbs getting a bad rap is not necessarily totally wrong, but I think understanding it as simple as you just ate too much sugar it's just too much. Like
Yeah.
it's not as simple as that.
Yeah. So some of the things you mentioned, just to echo some of that and riff for a minute on things other people have told me that I think of when we talk about this. So obviously there's intrinsic differences between people in terms of insulin secretion. At baseline, you're born a certain way, I'm born a certain way. You might release 15% more insulin than I do to the same bolus of food just because of intrinsic differences between us, say. There's also obviously what we eat. If either of us, eats more food in general or more of certain types of food, we're going to get more insulin release more of the time. That's going to come into play, especially if you're doing that chronically and at extremes. And there's also the issue of like endocrine disruptors. I've talked with people about microplastics like BPA, and my understanding is they're essentially exaggerating the insulin response if you get exposed to them at certain critical developmental time periods. This can lead to an exaggerated insulin response that sticks with you, right? So even if you're born with sort of a lower level of insulin secretion response to a bolus of food, if you're then exposed to an endocrine disruptor, it might
Hmm.
boost that, and then that will
Hmm.
predispose you to things later on. So you got all these things come into play here. Is it fair to say that a common denominator that's always there with type 2 diabetes is that for some reason or another, in some way, shape, or form, there was too much insulin release?
Um, so meaning you were making too much that caused the disease necessarily.
Making and secreting, I guess.
Yeah. Well, and that's what I mentioned. There are folks that think that hypersecretors or hyperactive beta cells might be a cause of insulin resistance and then eventually down the path towards obesity and diabetes.
Mm-hmm.
So, does that mean that if you had rested your beta cells this whole time, you would be better off, right? And so, I think that's where some of those beliefs come from. You mentioned, environmental, things that could maybe impair, either make your beta cells work worse and then push you over the edge to diabetes, or maybe early on make them hyperactive and maybe set off this insulin resistance cycle. I mean, I think that, the data on that is very much in real time and we're in our early days. If you're looking for guests to talk about that, I will plug Jenny Bruin, who's a scientist in Canada at Carleton University, who's doing like really, really elegant work understanding kind of, endocrine disruptors and environmental kind of poisons, to oversimplify it, that affect beta cells.
Mm-hmm.
But she's finding some really kind of interesting connections with these things. And I think, is it things like pollutants? Is it things like in our diet? There obviously could be many different things that we're being exposed to, and it's early days, but I think there are people definitely digging into that, and it'll be interesting to see
Mhm.
what the data and the evidence look like.
An area I really want to dig into here, because I know this gets into sort of the core of some of the stuff that you work on. Before we get to like your actual work, um, we're going to be talking about mitochondria here, I think. And I think mitochondria are becoming sort of a hot topic in many different ways across many fields, and I think that's a good thing. People seem to be appreciating them and getting interested in them more and more. Mitochondrial biology is fascinating. There's many things we could talk about. One of the, we'll just take it for granted. I've covered this on the podcast before. I think those listening will have some familiarity. Mitochondria used to be separate organisms that kind of got engulfed by bigger cells. So they're, in essence, little single celled organisms that live obligately now inside of our cells. But they have their own cell cycle and they still divide and stuff like that, even within our cells, I think. Can you talk to us a little bit about, in the context of beta cells, the mitochondrial biology of the mitochondria in beta cells? How are these things operating under normal conditions when someone is healthy?
Yeah, great question. Yeah, so it has been a little bit of a kind of revitalization in the mitochondrial biology world, and especially in the beta cell mitochondrial biology world. So, the old mitochondria, or the power plant, or the powerhouse of the cell, that kind of has been something you get in high school biology. It sticks with you forever. And then when I talk to people about what I do, they parrot it back to me, right?
Yeah, yeah,
Um, but what we're learning now is that these organelles are actually far more interesting than we thought, right? So they're not just client, or I call them client organelles that just make energy. Like they're more than that, right? They signal, they respond to the environment. They send signals to other organelles to, tell the cell what's going on, to maybe change cell fate and processes. And if we talk about our recent work there, then we can get into that. But as you mentioned, what's kind of the normal lifespan or function, or life cycle of a mitochondria.
yeah,
Uh, this is not work that I certainly like only found. This has been pioneered by many excellent groups, and actually in the beta cell community, probably the most prominent being folks like Niels Larson in Europe, and Orion Shearhigh and now at UCLA, and Klaus Volheim in Geneva. And so what we know is that mitochondria are not like a kidney bean that just stay the same way all the time.
yeah.
They undergo processes called biogenesis to kind of increase, kind of the biomass of the mitochondria.
So they
They
can literally
undergo,
grow and get bigger.
yeah, they grow, they expand their biomass, right? You don't like make de novo new mitochondria, but you can expand the biomass of the mitochondria, right?
Hmm.
And then these mitochondria live in networks. They're not like isolated little organelles. They live in networks, and the networks change. And so not all mitochondria are equal. There's a lot of heterogeneity. And so there's a process that's called fission that can separate the sicker, poorly functional ones out of the network
Hmm.
so that they can be cleared.
Hmm.
Um, and then if you get rid of those ones, then the network can reform through a process called fusion. And so this life cycle can go constantly. And when you reform a network through fusion, you can share metabolites, you can share nucleotides, you can share
Hmm.
DNA. Mitochondria have their own genome,
I see. So
right?
when you say network, there is a biochemical connection in the sense that mitochondria can sort of secrete and absorb things from other mitochondria.
Well, they fuse, right? So they fizz and fuse, they separate, they aggregate, and they change and evolve, right? And so, that's where it's not like, oh, if you look in an electron microscope, you can see a nucleus and you're like, oh, that's the nucleus.
Hmm.
But when you look at the mitochondria, they're not all the same. They're
Mm.
very different features, very different interactions. Some of them are really, really long and tubular and really big, and then some of them are short and stubby and circular, right? Because this is due to a kind of overall network related change in the mitochondria that's happening holistically in the cell. And beta cells do this too,
Hmm.
I think a unique thing about the mitochondria and beta cells are that beta cells are kind of not too dissimilar in terms of their mitochondrial biology. I don't want to speak too broadly here, but from a mitochondrial perspective, I think of beta cells, and I think they're underappreciated. They rely on their mitochondria not too dissimilarly from a cardiomyocyte or from a neuronal cell. And cardiomyocytes and neuronal cells are often thought of as like completely dependent on their mitochondria for their energetics and function. Beta cells are the same way. They don't really use glycolysis for energy generation. They primarily use glycolysis to make the building blocks to get into the mitochondria to
Mhm.
get the to keep the cell going.
Does that mean that beta cells are particularly energy intensive?
Oh yeah, absolutely. Because making insulin and secreting insulin is an energy intensive process, and these cells are, need to be rapid responders to metabolic shifts in the environment. So the mitochondria, the mitometabolism is really essential for that. I think one of the really strongest pieces of evidence for the importance of mitochondria in beta cells uniquely is there are these rare but notable heritable forms of mitochondrial diseases that folks get. And these are oftentimes problems in the mitochondrial genome. And the most there, but the patients with heritable mitochondrial disease, they have a lot of different phenotypes. They get musculoskeletal problems, they can get neural problems, they can get visual acuity problems. But the most penetrant thing that you see in mitochondrial DNA diseases is diabetes.
Hmm.
And it's not because they're obese.
Hmm.
These are folks that are born and get, diagnosed with these mitochondrial diseases, extremely early in life, right? So this isn't obesity. It's because their beta cells don't work
Hmm.
because of this inherited mitochondrial problem. So it just kind of highlights the real, like, crucial importance of mitos. And so they undergo this life cycle process because um, beta cells don't divide and grow very much. Again, not too dissimilar from a neuronal cell.
I see.
What you have at shortly after birth and within the first 6 months of life is pretty much what you're going to have for your lifespan.
I see. So these are long-lived cells.
Yeah, and long-lived cells really, really become reliant on their mitochondria to survive. But also the mitochondria need um, really efficient quality control processes to keep that cell alive for years.
Yeah,
Right.
because the cell isn't going to rejuvenate itself like, say, a skin cell constantly would. It needs to have a quality control mechanism for its mitochondria because it doesn't have the cell cycle to fall back on.
Right. Precisely. Precisely. Yeah. And
Interesting.
you hit the nail on the head. So, you can make, fibroblast cells that have really messed up mitochondria, and those cells may not grow great, but they can still grow, and it's because they can use glycolysis as an energy producer. A beta cell doesn't do that. It can't escape through the cell cycle to keep going. So
Hmm.
it's very dependent on
Hmm.
the upkeep of these healthy
I
mitos.
see. So where my mind is going here, based on what you've said so far, is these beta cells are long lived. Obviously, a big part of their purpose is to make and secrete insulin according to the demands of the body. And this insulin secreting and making process is intrinsically energy intensive. Therefore, this is my thinking here that I'm saying out loud. Therefore, anything that sort of stresses those beta cells, that's forcing them to produce more insulin more often, that's inherently going to be a very energy intensive process. And I would imagine that
Mhm.
doing that too much or doing that in a defective way is eventually going to break something, the same way that, I guess just running your car, with your pedal to the metal all the time is going to cause it to break faster than it would if you're following all the speed limits. So if insulin secretion and production are energy intensive and I am forcing a cell to constantly make more insulin, that's going to be highly, highly energy intensive. I could do that through eating more food, especially more of certain foods. I could do that through an endocrine disruptor getting in there and messing with the beta cells. And I imagine that could also happen just through some intrinsic inherited issue with the mitochondria of the beta cell. Does that sound reasonable?
It's definitely reasonable. And then this is where, the metabolic overload when you try to translate it to diabetes might not just be like, you had ice cream 3 days in a row, right? But
Hmm.
this is like decades of, progressive weight gain. And then these beta cells are working hard to keep up. And then the mitochondria are metabolizing all these nutrients and supporting the metabolism of the cell necessary to make the insulin to deal with the excess nutrients and also the potential insulin resistance, right? So it's kind of a little bit of a force multiplier
Yeah.
of you're doing an energy intensive process of the insulin production and release, but you're also dealing with the metabolites themselves, right? Then you can start to see how like, sick mitochondria in diabetes kind of can come into focus here.
Hmm. Hmm. When you transition from pre-diabetes to full-blown diabetes, is that a diagnostic line in the sand that we draw by convention, or is there actually some kind of like biological phase shift that cleanly demarcates the cells of a diabetic person from a pre-diabetic person? Is that or is it very much a blurry line there?
It's probably more of a blurry line than we would like. I mean, there are some distinguishing features, but I always remind everybody like the diagnosis of diabetes is like a construct, right? Like we say this blood sugar is too high or this hemoglobin A1C is too high, and then you have diabetes.
Hmm.
Now, those diagnostic numbers weren't pulled out of a hat. They were associated with like risk of microvascular complications and demise due to having sugars that high. So that's why we came up with those numbers. But there's a line that you draw, and then you say, okay, well, if you didn't go past that line, like what's going on with you then before? The pathophysiologic features are maybe a little bit more muted, but they're still there, right? And you know the there are again emerging studies. There's not a ton out there on studying beta cells in the pre-diabetic phase, mostly because when we get islets from a deceased islet donor, first of all, this is a tragedy, right? Somebody's died. Um, we don't get if someone doesn't have diabetes, we just hear that they don't have diabetes.
Hmm.
And so then we have to make some assumptions, and all of our deceased donors have this glucose, they have glucose levels measured, and then they also have the hemoglobin A1C, which is look at your glucose over a 30-day period. And so when we call a islet donor pre-diabetic, it's kind of artificially based on that hemoglobin A1C value. But we don't know like what was going on in the 3 months before the person died. Like, were they really on their way to get diabetes, or did they have like something change? Were they admitted to the hospital for a few months and being pumped full of medications and
Yeah,
steroids and stuff
yeah.
that so why am I mentioning this? The reason I'm mentioning this is when we get an islet donor with diabetes, you know, the the physicians that have prepared the reports, they say, oh, this patient had diabetes, they took these meds. Like, this. But when we try to do molecular studies of pre-diabetes, it's a little bit of guesswork because very rarely do people walk around with that diagnosis of pre-diabetes. We're doing a little better as a clinical field at doing that now, but it's certainly not something like regularly reported. So when we get these islets and we call them pre-diabetes, it's often by stratifying their hemoglobin A1C and then seeing if they mirror molecular changes that you see in type 2. And there are a lot of similarities, but they're often a little bit muted. One thing that we do see is evidence of, transcriptional evidence of mitochondrial problems in these beta cells. Does that mean that, you know, like had we looked even earlier, would we find them? I don't know, right? And I think this is the thing that we are really trying to do as a field, the islet biology field, is like, what is the what are the initiating events? How early can we go to find things beyond just assessing the genetics in a static way to say,
Mhm.
oh, you have these genetic risks that could give rise to mito dysfunction or generalized beta to cell dysfunction. But like, what is that pathologic change that happened? right now we're very reliant on these pre-diabetic,
Mhm.
islets uh, based on assumptions from the A1C.
Mhm. For someone who has type 2 diabetes, and let's just imagine the average person with type 2 diabetes, whatever exactly that means, there's probably, right, some extended period of progression here. It's not like they wake up one day and they simply have it when they didn't have it the day before. There's a phase of not being diabetic and then being pre-diabetic, and then eventually they meet the diagnostic criteria for type 2 diabetes. At key time points along that trajectory, what does reversibility look like? Is it mostly reversible for much of that timeline? Is it partially reversible? Does it at some point become totally irreversible?
Um, I think that reversibility would probably be dependent on something that is you can kind of measure easily, and then something that's a little bit more difficult to measure. But I think that if you lose your beta cell reserve, the reversibility becomes much lower. The possibilities go down. Now, we don't often do a lot of dynamic testing of beta cell function in the clinics, just mostly because, some of these tests aren't standardized, and then like it's hard to often interpret like, oh, does that mean you have like 80% capacity, 60%, 40%? We use these in clinical trials, but we don't always use them in clinical practice because it's hard to translate to the population. But I think if you look at folks that can do, again, now we're in this era of Ozempic and Zepbound and these things, and you can really promote substantial weight loss and dietary modifications, there are going to be people that probably aren't going to ever be cured of their diabetes from those things because their reserve is just not there, or not remediable. And is it either that their secretion just doesn't come back, or the beta cells were just lost and they're just not
Yeah.
around anymore?
And it could be either to some extent. Is that what I'm hearing?
Yeah,
Yeah.
yeah. And it's mostly because we just don't we don't know a lot. But again, it is possible, right? Because certainly, that study that I cited a little ways back, if you take, there's especially early onset T2D, if you do really, aggressive diet management, there half of them you can get their blood sugars into the non-diabetic range. So there is
Mhm.
some plasticity of the process
Right.
to
Right.
come back.
And what's sort of the common denominator there? So when you're type 2 diabetic or pre-diabetic and reversibility is possible, and it's been seen in some studies, what is the diet management there? Is this just focusing on getting insulin as low as possible?
So I think that's I will again plead my ignorance to my colleagues in the field who are looking into like the dietary approaches. I I will say, to simplify it, it's certainly about caloric restriction is key. And I think reducing your calorie intake is important. Now, are there some diets that may be more advantageous than others? Like when I was coming up through my training, and things like Atkins and South Beach diets were like really popular. Actually, even before that, Mediterranean diets were the thing. And then now, I think we have different flavors and permutations that are all kind of around that. But I mean, if you look at largely those diets, they're again lower calorie, lower carbohydrate, lean protein, vegetables, those types of things. I think those, instead of looking, I know a lot of times we always want to look for like the magic bullet, but I often am like, what is the overlap in the Venn diagram?
Mm-hmm.
Because then that's probably the thing most applicable to everybody, right? Whereas like maybe one diet will work for one person. Oh, if you do Atkins, you just don't eat a carb the rest of your life, you're going to be fine. But for some people, it might not work. And so that's why I think that, pretty smart caloric restriction and reduced carb intake seem to be very safe ways to go. In terms of lowering insulin levels, um, you know, I think it's reasonable to think if you lower insulin that as an ambient readout of peripheral insulin resistance, it's a good thing. When I'm in practice now and I'm seeing a patient, though, it's again, there's not a lot of standardization. So, like, I
Mm-hmm.
can't bring them in and say, I can measure their serum insulin levels the day they come in, but it could be variable. And I might not be able to say, oh, you're at like the magic number now,
Mm-hmm.
right? So we haven't gotten there yet. We have continuous glucose monitors. I don't know, maybe we'll eventually have continuous insulin monitors. And then we could get really, really granular on like your insulin levels need to be this to really see effective weight loss. We're probably not there yet, but it's not outlandish to kind of think, in the abstract in the future that like lowering food intake and then some way to monitor your insulin resistance could be a way to say, oh, look, you're unlocking these options that could help you remediate the disease.
Mhm. What can you tell us about some of the potentially intrinsic beta cell problems that you might see in someone who gets type 2 diabetes, even though they're not obese, and I mean, just something intrinsic that might be born with. Is there, and to the extent that's there, like, what is it? Does it have to do with the mitochondria? Does it have to do with just an inefficiency in secreting insulin that person has from the get-go?
Yeah, so there's a lot of discussion on like what are the cardinal features of like a beta cell that is kind of on its way
Yeah.
down. And I think the ones that seem to be most commonly seen are, there's definitely a component of mitochondrial dysfunction, and obviously that's what we study. So it's in my lab, so we're pretty up to speed on that. There has been, I think, about 20 years of focus, and some nice data on the role of endoplasmic reticulum stress as a component. And again, is it an initiator? Is it responding to something else? There's people that debate those things, but thoughts that, the ER are where a lot of the initial insulin processing and folding events and, occur. And so if you're a beta cell and your job is to make and release insulin, you could imagine that like problems in the processing and generation process in the ER could be pretty fundamentally bad. So there's components of that. Um, the two other things I think that you see, and by the way, I'll mention two other things, but a lot of these things could also be interrelated, right? There may not be mutually exclusive. Two other things that you sometimes see are problems with a process called autophagy,
Mm.
which is another cellular regulatory process to kind of eliminate, sick or damaged organelles, or cellular structures or aged proteins. That seems to kind of slow down and be impaired. And then another feature that, has been reported, and again, some debate on how important is it, but I think, are regularly seen, is the deposition of amyloid in the beta cells of a type 2 diabetic. And I know you'll say amyloid, you'll think about Alzheimer's disease, but beta cells make a protein called islet amyloid polypeptide, which bears some similarity to the amyloid in the brain, but also some distinguishing features. And IAPP is co-secreted with insulin. It's made, it's processed in beta cells too. And it can form aggregates, and those amyloid aggregates can also be toxic. So, you put some of those features together, and then you start to see, there's a multi-hit approach starting to occur that's damaging your energetic potential of the cell that you need to kind of feed all these processes, eliminating your ability to get rid of kind of sick and old and not functional stuff through autophagy. You're depositing aggregates in cells that probably are also nidus for things getting bad. By the way, if your autophagy is not working, you can't get rid of those aggregates, those
Hmm.
amyloid aggregates. And then fundamentally, the ER, which are their job is to make and process insulin, the ER also are not really up to snuff as well. And so you have like a lot of these kind of hits occurring
Hmm.
that then ultimately sometimes feed back to the nucleus and then affect kind of the whole transcriptional nuclear program in the cell too, that may affect cell survival and fate.
Yeah, yeah. I'm struck by what you told us earlier that these pancreatic beta cells, so they secrete insulin. It's very energy intensive. These are long-lived cells. So it sounds like there's an analogy here that I think you mentioned between these pancreatic beta cells and cells such as heart cells, and neurons, long lived cells that essentially never divide or very rarely divide. And as a consequence of not being able to do a cell division to rejuvenate themselves, they are dependent and also vulnerable to certain bioenergetic disruptions that other cells might not be.
Hmm. Hmm. Hmm.
And you just mentioned that you can get some of these beta amyloid like tangles that build up in these pancreatic beta cells. So is there a connection here where long lived cells that are not subject to the same sort of recycling mechanisms that can rejuvenate other cells are vulnerable to types of cellular degeneration that other cells just aren't?
Yeah, I think that, they're just these are cells that for whatever the reasons seem to have certain kind of proclivities and sensitivities to certain things that other cells may not encounter, may not see, or are far better equipped to deal with, right? So, we talked about the analogies with amyloid and the like, and there are like patients with Alzheimer's or with Parkinson's disease are at a higher risk for developing diabetes.
Hmm.
And patients with diabetes are at a higher risk for developing those neurodegenerative disease. Are they all caused by the same root cause? Like, I'm not going that far. Are there shared pathologies? Possibly. Is it are they genetically the same? I don't, again, I wouldn't go that far. But I think it just speaks to if these things are related in some way, it may just speak to the fact that there are certain cell types in the body that seem to not be able to particularly well withstand similar types of stressors.
Mm-hmm. Mm-hmm.
Um, and their long-lived nature is likely a very big part of it.
Yeah, yeah. If from a clinical perspective here, you see patients. So if, let's say you have someone come into the clinic and they're either pre-diabetic or even they're healthy, but in either case, they're very motivated. They don't want to become diabetic. They want to reverse their pre-diabetes or just stay healthy and not become even pre-diabetic. What are some of the key pieces of practical advice that you think apply to most people most of the time? Would
Mhm.
one of them be, get a glucose monitor and make sure you're not spiking very high and very low as often as possible? Would you make any sort of umbrella? Is there any umbrella advice when it comes to specific dietary and lifestyle interventions, or what would you tell someone who's interested in
Yeah.
in preventing diabetes?
Yeah, 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 yeah. It's a great question. So I wish I had more of these patients. Unfortunately, because of the needs in our clinics, like I get the people with diabetes, not the ones that don't want to get it. But sometimes they come with family members that don't want to
Yeah.
get it. So um, you know, I don't regularly advise folks go out and get a continuous glucose monitor just because it's hard to know what to do with that information, and maybe we will be able to equip that information and learn about how to utilize it effectively in our patient populations that don't have diabetes or might be our at-risk patients in the future. So I would say right now today, you may not gain a lot of value out of buying one, other than you'll see the numbers and maybe it will, like when you have that helping of ice cream, you'll go, oh, my sugar went up. Look at it went up. Maybe I shouldn't eat that ice cream anymore. But in terms of like actionable information of like, we're gonna follow this tracing and say this is bad and you're at risk. I don't know if we're there yet with the continuous glucose monitors, but the story will still be told as we apply more technology and more importantly, as the technology gets less expensive, right? Um, so I think then, we get into practical things for folks. And, look, while I did say that if you were overweight, 4 out of 5 overweight folks don't get diabetes, 1 out of 5 still do, and 20% risk is still not ideal, right? You wouldn't want to play with a risk for a disease that could really affect your lifespan and is carries pretty substantial risk of death due to cardiovascular disease. So the number one thing I would say to somebody who comes in is, do everything you can to stay at a healthy body weight. And what does that mean? Exercise, eat right, stay active, watch your caloric intake and those things. Those are, I think, and you might say, well, of course, like Captain Obvious, but like, those things really, I think, matter here because I think you're minimizing the risk to straining your beta cells and you're minimizing the risk to those kind of that cascade of things that may lead to diabetes later. So that's kind of priority one, as I would put it, is dietary management, weight management, and then staying healthy and fit and exercising. Is it
Mhm.
going to prevent you completely from getting diabetes? Of course not.
Mm.
There are many folks that will still get
So,
type 2
yeah,
diabetes at a relatively healthy body weight.
yeah.
So, but it
So let's
certainly can lower your risk a lot.
Okay, so let's say you have patients come in. I mean, imagine you have this at some rate. You have patients come in, they take your advice. They say, I've been exercising more. I think I've been eating right. I feel like I've been eating less. I feel hungry more often, but they're struggling to lose weight and nothing seems to be improving. What's sort of the next layer of advice?
Yeah, so I mean, this is the challenge, right? So I think if you're someone who's overweight and you're doing everything you can from traditional dietary methods and exercise methods, you know, again, you may be someone who's adipocytes and body weight, you're just, you don't, you have a higher tendency to gain and hold on to body weight. Your body weight set point might be higher. And certainly that's, we're in this era with, GLP-1 receptor agonists and tirzepatide and these dual receptor agonists that are coming on the market to try to assist for people that, despite really kind of sticking with it and doing the best they can for weight loss, don't get there. And certainly, then it's a decision between you and your physician, but it's understandable. And certainly we have people that, and now the FDA has approved these drugs for obesity independent of diabetes, right? For this reason. So medical weight loss is an option. Structured dietary programs are an option. We have an intensive weight management clinic here at U of M that it's not just come in and get Ozempic and Zepbound and Wegovy. There's a dietary component to it. And, for the dietary program, they're providing like pre-packaged, meals to the patients. So, because sometimes it's like, go do this diet, but then it's like, well, then you're left to your own devices. And did I do
Do
this?
you know what's in those
Whatever.
prepackaged meals?
Right. So, I mean, and I'm not saying that like people want to eat that way, but sometimes it really helps when you have a challenge. So, so we have an intensive weight management program that provides that support too, right? So that you're really staying on the caloric restriction to the best of your ability. But it's not always like a one size fit all. So, right. So I think, I look at it as you need to consider the all of the above. And then let's not forget, although, now in the era of medical weight loss, we're not thinking about it as much, but there are surgical options, right? Bariatric surgery is still available. A lot of people still pursue that. It's very effective. You have newer things like endobarrier and band and these things too. So, there's a better and more comprehensive toolkit than say there was 30 years ago, 35 years ago when it was just exercise and diet. And if you just didn't have if you weren't dealt the right genetic cards, then, you weren't going to lose weight, right? So I think that, you have to consider that it's nice that we have choices and then hopefully we'll continue to have more choices in that way so that we can find fits because certain things are going to work for people better than others.
Hmm. What about someone with type 2 diabetes? How do you talk to them about using and looking at the continuous glucose monitor data?
Yeah, well, so when you have type 2 diabetes, then obviously that data is really important to canvass their glucose control. So that's really given us a lot of powerful information to learn about overall glucose control. In the old days, if you're doing finger sticks, even if, and I was a person that was doing between 4 and 8 to 9 a day, and somebody would say, geez, 7, 8, 9 finger sticks, that's so many. Um, But when I did those that many finger sticks, or even four, which was what we used to recommend for type ones on insulin as the minimum, um, that means that during basically 4 seconds, the second that your meter reads to you, a day is the only 4 seconds that you know what your blood sugar is, right? So now think about it. Now you can see what your sugar is like all the time, right?
Hmm.
So it's really powerful. It can tell you, when patients have high sugars. What are the irritants to their sugars? Oh, hey, you were doing well at breakfast and lunch, and then your post-dinner numbers skyrocket. What was happening then? Um, oh, you wake up in the morning super high. Oh, do we have to think about counter-regulatory hormones that are at play here that are, normal hormones that help you wake up but counteract insulin? So it just gives a picture so you can structure therapy in a much more logical way. Um, and also allows us to talk to our patients about, you know, rather than once every 3 months hemoglobin A1C or looking at a glucose log to say, you're doing well, you're doing kind of well, you're doing excellent, you're doing poorly. Like you can grade things out in a really granular way to try to chip away at problems with blood sugar. So very important to have that CGM for diabetes. And of course, you need insurance to cover it in the US. So, we keep crossing our fingers that, insurance providers and Medicare, Medicaid, support these things because they make such a difference.
Hmm. Um, we've already covered a lot. Is there anything that you think we haven't covered off on, or anything you want to reiterate about what we did talk about? Final thoughts that you want to leave people with?
No, I would say this, and I kind of leveraged it at the beginning, but like this is not like widely held belief. This is a little bit more like me maybe riffing and giving a little personal opinion. But I really think that diabetes is on a continuum.
Mhm.
And I think that the more, there is this desire to subclassify the forms of diabetes because it's like, oh, if we develop more classifications, we'll learn more, and then maybe we can do personalized therapies. Some therapies will work better in some forms of diabetes than others, have different risks. I'm 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 not in any way opposed to some of those things if they're actionable in the clinic. But I think holistically, the thing that I really believe is that the continuum of diabetes has something to do with a beta cell problem and then these irritants, autoimmunity, obesity, metabolic overload, inflammation that come at this. And the one thing that we have a lot of great treatments for diabetes, and hopefully we're going to have better. We don't have treatments that rejuvenate and make your beta cells healthy.
Mm. So
And to
we
me,
know that's
I think
we
that
know
is
that's a
the
key.
next frontier.
Okay. So yeah, we're not going to have time to get into like details on the medications, but it sounds like what you're saying is that beta cell dysfunction's at the core of this, and for better or for worse, the medications and the treatments we have today for diabetes don't actually address that piece.
No, I mean, the best that they do is offload work on a tired or sick, especially in type 2, a tired and sick beta cell, um, so that it can kind of limp along or maybe recover,
Mhm.
or especially if you lose a lot of weight, maybe recover to the point where your blood sugars are almost well controlled. Um, but there is no medicine that I can give you that will rejuvenate your beta cells. There were some very nice clinical trials that looked at some of the most commonly prescribed diabetes therapies, and the only one that seemed to preserve didn't enhance, preserve beta cell function in the long term was surprisingly drugs in the TZD class. This is drugs like Actos
What is
that are
that?
very rarely prescribed. These are PPAR gamma agonists, right?
Mm.
We very rarely prescribe those medications now,
Why?
and they're called insulin sensitizers because they are. But actually, that was surprising. That was the only drug class that preserved beta cell function. Um, None of the other drugs do that.
Wait, so why aren't those ones more widely used? Is there some other reason?
So a little bit of adverse effects and then a little bit of bad press too. I mean, in about 15, 20 years ago, there were some, I don't, again, I don't want to go back and relitigate it, but like there were associations drawn between Avandia and heart disease. Was that very strong conclusion? Who knows? But
Oh, I see. So the
immediately,
drug companies basically got spooked.
well, there was a lot of spooking, and then prescribers got spooked too, and so then you saw a precipitous decline in the use of Avandia is no longer on the market.
Huh.
More recently, there have been some suggestions that TZDs may have other off-target effects that might not be great, but like most of the time, people don't use them because there can be a little bit of weight gain.
I
It's
see.
like fluid retention weight gain.
I see. I see.
Um, and and so but it's largely driven by just like some, just a little bit of the field getting spooked from using them.
Hmm.
And it's so they're no longer early, used to be like a very early line agent. Now it's kind of like third line, fourth line agent that you use TCDs. And I'm certainly not here going like everyone should be on one, but one of the things that I thought was fascinating about the TZD literature is that it was the only drug that seemed to preserve and sustain beta cell function in type 2 diabetes. And so it just reiterates this point for me, which is we don't have anything to preserve beta cells. And if we did, I wonder about our long-term ability to make really impactful things for people with diabetes. And
Hmm.
it's not for lack of trying. We don't have that magic thing, and there are people working on candidates to do that, but it remains a really substantial challenge is finding drugs that rejuvenate or regenerate beta cells or offload the stress of these beta cells so that they can recover.
All right. Well, definitely learned a lot in this one. Thanks for covering this topic. Thanks for reading with me. Really appreciate it. If there's nothing else you want to leave people with, I think that's a good place to end it.
Oh, great. Yeah, thank you so much. And I'll just want to plug that, I work here at U of M, the Caswell Diabetes Institute. For folks in the Midwest that are seeking diabetes care, for students and trainees that want to work in a really vibrant diabetes environment. This is kind of like, I think it's the Midwestern. I grew up in the Midwest, but I spent 12 years on the East Coast. We're not great at trumpeting what we have available, but this is probably one of the more comprehensive diabetes ecosystems in the country for clinical care and training. And so I would tell folks if you're interested in learning more about diabetes and training in diabetes, consider coming here. Or if you're someone with diabetes who wants fantastic care, that it's a real passion of not just me, but our colleagues. And we're really proud of what we do in the research and clinical space here.